Thank you for the introduction and thank you also for the invitation, despite being not in the CRITEX project. Actually, the Econect project aimed at designing an observatory with different measures and different compartments of the ecosystems, but mainly, as Jerome said, on living organisms used as sentinels of the environment. So why is it important to monitor living organisms in ecosystem? Of course, living organisms play a major role in ecosystem functioning. We cannot understand most of elemental cycles without considering this living organism. This is the same to build matter or energy budgets. And also the role of this organism is pivotal for ecosystem services provided to the society. Living organisms are strongly impacted by environmental changes, mostly human-driven. And in this context, getting relevant information about their population dynamics is an important issue, for instance, to build local or global trend indicators. Finally, living organisms are also bring information about environmental properties. For instance, here I display some pictures showing how the identity and the abundance of organisms are changing during the eutrophication of shallow lakes. And the whole community or some single species can thus be used also for bioindication purpose. This brings me to the concept of sentinel species, which is defined as any organism, so most often animals, that can be used to detect risks to humans by providing an early warning about a danger, in particular in the context of environmental hazard. And to illustrate this concept, I like showing this picture taken in the 1920s, on which we see two miners with a caged canary. As you know, canaries and other bird species were used in mines to detect carbon monoxide due to their high sensitivity to this gas, much higher than humans. Of course, today we have accurate electrochemical sensors to measure carbon monoxide concentrations. And more generally, the interest of sentinel species is not to get information on a single parameter, but it lies in their capacity of integration, which is particularly useful and important in a multi-stress context. Also, the current technologies, including IoT and artificial intelligence, open new perspectives for remote monitoring of sentinel species and for the real-time analysis of the data collected. When we speak of automatic monitoring of living organisms, there are basically two main strategies. You can equip animals with various sensors. It's what we call biotelemetry or biologging. And the fact to directly instrument animals offers many possibilities to study the animal itself or its environment. But it is also quite challenging, especially for the smallest species, because you have to embed all functionalities of measuring instruments in a small volume and with a low weight. An alternative is to install fixed stations collecting data about surrounding organisms. One of the most known measures is the passive recording of sounds with commercial devices such as this audiomorph system you can see here, or with more sophisticated devices such as this one used in the Australian Acoustic Observatory. Another well-known approach is the camera trapping, and in each of these cases you get some information of the presence of various animal species. So I'm going to concentrate my talk on fixed observations, and you will get that in addition to images and sounds, we can get many other information as well. Before setting up fixed observations, observatories to get observation on living organisms, and thus before getting data and using it to do science, there are a few questions to answer. First, which metrics should we use to monitor living organisms? These metrics should, of course, be informative, and their measure should be as reproducible as possible to provide standard observations. These standard observations will then allow to compare, of course, among sites and over time. You cannot measure everything with sensors, so you will have to go for automatable metrics. And however, the fact that a metric is not automatable at one moment, notably with commercially available sensors, does not mean it will never be. Things are evolving quite fast, and the creativity of researchers and engineers can open new horizons. Also, the willing to perform observation in natura within a network of remote stations implies, as Laurent has reminded just before, to use robust, weather-resistant instruments. Power supply is often also an issue, and of course you can really take advantage of remote observation only if you can upload the data, some of them at least, and ideally directly on the researcher's desk. However, when you deal with large datasets, this may be relevant to analyze them automatically as close as possible to the sensors. This is what we call edge computing, and this allows to upload only useful data. For instance, when you deal with animal images or sounds, it would be a good option to not transfer the raw data, but only the time-stamped occurrence of the species. So technology brings a lot of promises, but we should not forget some ethical issues associated to the automatic monitoring of the environment. Even if ecosystem instrumentation does not weigh much compared to the recreational applications of connected objects, its use is increasing, and it is important to question its possible impact on the environment. So we should consider also such impacts relatively to the benefits of the monitoring conducted. Of course, there is a temptation to consider that ecosystem observation is inherently useful, but this is not that simple. So we have to carefully consider our strategies of development of new sensors and of their deployment in nature. The Econect project is a 3-year project that will finish at the end of this year. So some of the results I will present are quite preliminary. It involves 6 research labs, both in environmental and technological science, and 3 private companies. It has been financed with European funds brought by the Occitania region. The objectives of the Econect project are first to develop a shared architecture to collect and analyze environmental data, then to apply this architecture to 3 sentinel species of the environment: a common passerine bird, the great tit, a pollinator insect, the bee, the honeybee, and an aquatic invertebrate, the pond snake. Then to deploy a network of measuring stations in French Occitania, and finally to use the data collected to assess the relevance of these sentinel systems to study the effects of human-induced pressures. This is the architecture of a typical Econect measuring station. On each site we have different sentinel species, such as nest boxes, bird feeders, beehives, and aquatic observers. All these devices work autonomously and have communication abilities to transfer the data they collect to a local communication gateway. The gateway will then aggregate the different data and send them to a remote server where data are processed and where they can be visualized by the researchers, of course, but also by citizens who will have access to pre-interpreted data. Citizens also play an active role in the observation process, notably by contributing to collect additional data and to maintain the devices on site. And beyond the help they provide, this is a good opportunity also to involve them in some environmental educational programs. So in the Econect project, we have set up a network of 12 study sites across French Occitania. These sites have been selected to illustrate various bioclimatic conditions and contrasting land uses. 5 sites are mainly forested. The disc you see here have a radius of 3 kilometers, which corresponds more or less to the foraging distance of a bee. 4 sites are mainly agricultural, with a gradient of intensification of practices. 2 sites are urban, within large agglomerations in Toulouse and Montpellier. And the last site, with an heterogeneous landscape of scrubland and crops, is located close to the former ore mine of Salsigne, one of the most polluted sites in France, mainly by arsenic. All of the sites are being characterized in terms of chemical contamination by measuring trace metals and pesticides in various environmental matrices. I'm going to present now the different Sentinel systems used in the project, starting with those for monitoring birds. So this is the Smart Nest, which is a connected nest box designed to fulfill the requirement of a great tit. The system is based on Raspberry Pi Zero with a night vision camera. We also have an infrared barrier to monitor the activity of parents by counting their entries and exits, and sensors to monitor temperature inside and outside the nest. The pictures are sent by the Zigbee protocol to the local gateway. Here you have some examples of pictures taken by the Smart Nest. You can see here 8 chicks shown at different stages of development. What you see here is the first version of a connected bird feeder. Actually, it is much more than a bird feeder. It is a device to measure cognitive abilities of birds in the wild, or in other words, to perform IQ tests on birds. The access to food is prevented by a small actionable shutter that can be opened when a bird is detected on the perch by its RFID chip. However, the device is also equipped with some little colored light diodes. You cannot see them really here, but they are here on the top. And only when there is a certain light stimulus, the shutter can be opened. After some time, the birds can learn the association between the light stimulus and the food reward. And on this graph, you can see the learning curves of 3 bird species. So you can see that the blue tit is the faster learner, followed by the grey tit, and finally the march tit. This should be noted that for a given species, there is also some variability due to environmental conditions. And for instance, urban birds learn faster than rural birds. Next step for us, testing on Econect stations the influence of chemical pollution on these learning abilities. Here you have the last version of the open feeder. Among the main changes, we have a video camera and this articulated arm that could be used to individually recognize birds for the cognition test. But we are also considering the possibility to adapt cognitive metrics at a population level to overcome the need to individually identify birds. That said, even if we do not need individual identification, the camera is still useful at a community level to identify bird species and then to monitor the species richness and the relative abundance of wintering granivorous birds. Let's see how we can do it automatically. We have worked to develop artificial intelligence algorithms based on deep learning using convolutional neural networks for the identifications, so not of cats and dogs here, but for bird species from the frames of a video stream. Our purpose was to get detection algorithms, so they first delimit bounding boxes around the elements of interest, and then identify them, whatever their position or number on the picture. As you can see here, you may expect birds and get some other animals. This should be accounted for when developing the algorithm. I believe this is useful to say a few words about the way to build and train such algorithm, even if I'm not going in details to the techniques. What we did is called transfer learning. This means that we used a general purpose computer vision algorithm already trained on a massive dataset, several millions of pictures. In the present case, it was the MobileNet V2 model. And then we only trained the last few layers of the neural network to adapt the model to our use case, namely 13 classes of birds and other animals attending the feeder. For the transfer learning, we used a bit more than 30,000 of pre-labeled images. Half of them have been collected on the Econect sites with a device we specially designed for this purpose. And the other half was obtained from the web database eNaturalist. So in other words, we used on average a bit more than 2,000 images per species to identify, which is not so much. But how efficient is it? The accuracy of our model, which is the ratio between the sum of a positive and true, of true positive, and true negative on the total number of tests, is 87.5%. But what about other projects in the literature? Is the use of convolutional neural networks for species identification widespread? And is the average accuracy good enough to allow their routine use for biodiversity monitoring? To answer this question, we performed a meta-analysis and we found almost 500 publications using convolutional neural networks for this purpose. As you can see, since 2009, the number of publications has increased quite fast. The first mention of embedded system also was published in 2018. And globally, the average accuracy of the different studies providing usable results is 86%, which is quite good and pretty similar to the accuracy of our own model. We also examined which factor could explain the variability in algorithm accuracy. As you can see from this graph, accuracy is notably influenced by the number of images per class, but in line with our own work, 1,000 or 2,000 images is enough to get proper results. Finally, we also address the question of environmental impact of automatic identification using smart cam traps compared to field operators monitoring various components of biodiversity. So please note that these results are only preliminary and the comparison here concerned only the carbon emissions. So for the human approach, we calculated carbon emissions from the information sent by 45 respondents to a server and based on questionnaires. And we notably accounted for car travels for the use of numerical tools to analyze and to store the data. For the automated approach, we considered a network of 10 smart camera traps, and we accounted both for the emissions associated to the device manufacturing, for the training of neural networks, for the energy costs associated to the functioning of devices, and to their installation in the field. These are the results we obtained. The exploitation of these results is not straightforward, as we compare two monitoring strategies that are really different. On the one hand, the cam trap approach allows to monitor only a small area, 300 square meters per device, and at the opposite, this is done continuously, night and day. In contrast, for field operators, they investigate on average 8,000 square kilometers per year, but do it in 40 hours. So if you compare the two approaches based on time units, the automated approach is emitting less carbon. If we compare in terms of surface investigated, in contrast, the human approach seems to be much less emitting. Of course, we must be careful about these results, and as we calculated average values on a variety of situations for field monitoring by operators, and also carbon emissions are not the only environmental impact associated to automated approaches. We should also consider toxic emissions at the various stages of the life cycle. And last, there are other drawbacks of automated monitoring that should not be forgotten. For instance, the disconnection from the environment they could generate by comparison with human direct observation. Let's consider now the connected beehive that you can see here. This hive is notably equipped with sensors to measure outside and inside meteorological parameters. Sensors also to monitor the weight of the hive, which depends both on the importance of the colony and on its food storage, and also sensors to count the number of bees entering and leaving the hive. These are the kind of time series we record. Here you see the dynamics of a given hive in spring. On these curves you see inside temperature in blue and outside temperature in red. You can see that even during cold episodes, bees, at least a healthy colony, can maintain its temperature quite stable. On this curve, you see the evolution of the hive weight beyond circadian variations that in itself can bring information. For instance, you see here that we have different patterns during the cold episode. You can see that there are some day-to-day changes that can be quite fast in weight. For instance, here, and this was correlated with flowering of robinia, which is a tree species, of course, producing a lot of nectar. The next step will be to be able to automatically analyze these various time series all together, as this was shown in the previous talk, also to get integrative vision, in our case, of the colony dynamics. Although the metrics mentioned in the previous slides already provide relevant information about colony dynamics, we aimed at having a fine and fast response in case of brain dysfunction, as can be caused by some chemical pollutions, for instance. There is indeed a major challenge in detecting these weak signals that can announce coming collapse of a colony, but quite some time before it happens. And it can announce also some important impacts on the communities of wild pollinators. Of course, it's much more easy to monitor a hive with domestic bees, with honeybees, but of course it's also an umbrella species that gives information on all the wild pollinators. The initial project was to design a connected flower. Well, I must admit it does not really look like a flower, but still the aim to study bee cognition is reached with this device. So the functioning is similar to what I have shown for the bird feeder. Bees are individually tested in this kind of maze you see here. And once the bee has entered, it can choose between two colored lights, and only one is associated to a food reward. Then the performance of individually tagged bees can be monitored through their successive passes in the device, all the process being automatically controlled via computer vision algorithms. These are the kind of results we obtain. I'm sorry, the quality of the graphs is not so good when it's displayed. Showing so here on the left the percentage of individuals being able to do the right choice in function of a number of attempts. And similarly, we can register the evolution of the time taken by individual bees to get the food reward. Some other results, but with a reversal of the rules of the game in the middle of the experiment. You can see that the bees are quite fast to accommodate to the new rule. And this approach can provide complementary cognitive metrics because the neural basis of initial learning and of reversal learning are not the same. Here you see the final device developed by Bigward in the frame of Econect. I come to the last Sentinel system, the Aquatic Observer, which is a floating platform with communication abilities and energy autonomy, thanks to its solar panel. This system can host different measurement devices, notably to measure water physicochemistry. This system is based on the open hardware Arduino Promega board, and its various buses here are used for communication modules and for probes and sensors. Within this version of the device, we measure conductivity in water, so also in water pH, redox potential, distilled oxygen, temperature, and also ambient light above the water surface. But an important characteristic of this instrument is its versatility. It is absolutely not a problem to add new sensors, either commercially available or developed in the lab. So when we want to increase the replication of the measures, and also if we want to share the approach with citizens that could contribute maybe at one moment to these measures, the cost of the device is an important issue. So what is the cost of our Aquatic Sentinel? It's a bit more than 1,000 euros as a whole, with components all available off the shelf. As you can see, most of the price comes from probes and sensors. And this cost could be even lowered by developing new sensors, as our colleagues at the LAS, for instance, are currently doing. Also, our Sentinel can be defined in various versions, and if solar autonomy is not necessary, of course, this is another way to save money, as well as to reduce the size of the device. Even if our system is already allowing to indirectly address the health of aquatic biota, for instance, via the circadian fluctuations of dissolved oxygen, giving information on the metabolism of the ecosystem, we examine the possibility to add a living Sentinel on board. For that purpose, we first develop a computer vision algorithm able to track the movements of the pond snail, which is an invertebrate species often used as an ecotox model. In parallel, in the lab, we examined how relevant the locomotion activity of this snail is to detect chemical contamination. For this purpose, we made a test with copper at various concentrations from 0 to 40 micrograms per liter. As you can see, the test was quite demonstrative, with a significant decrease in locomotion activity for concentrations as low as 10 micrograms of copper per liter. As a comparison, you may note that the legal standard for drinking water is 1,000 micrograms per liter. This high sensitivity of a behavioral metric is not really surprising. In our group, and also what we found in the literature, in the ecotox literature, has convinced us that both behavioral and cognitive metrics are often more sensitive than endpoints based on physiological biomarkers, for instance. So this makes behavioral metrics ideal to get early warning of chemical contamination. To finish with our Sentinel systems, just a few pictures about prototype for including snail monitoring in our aquatic observer. So by the way, this is something already done in different contexts with other aquatic invertebrate species by some ecotoxicologist colleagues, also in Lyon, in Bordeaux, or in Le Havre, to cite only French groups, and which is sometimes called e-caging or smart caging. So the animal is directly in the environment, and then its locomotion response or other behavioral metrics can be used to get a response to the fluctuations of the environmental parameters. So I spent quite some time on the Econect Sentinel systems because I really wanted to show you the potential of automated fixed observation to take a new look on living organisms and also on the meaningful ecological information they convey. But I will now end my talk with a few slides to show how we deal in the Econect project with the massive data produced by our Sentinels. As indicated, all the data are locally aggregated by a gateway, This gateway is based on a Jetson Nano board, which is similar to Raspberry boards for people knowing this kind of technology, but with extensive embedded artificial intelligence capabilities. The Econect gateway is compliant with several communication protocols. So the LoRa protocol is ideal, for instance, to get data from Sentinel devices at distances that can reach several kilometers with a very low power consumption, but they are limited to small volumes of data. So LoRa cannot be used to upload sounds or images, for instance. On the contrary, the Zigbee protocol can transfer images. It's what we used for the Smart Nest, for instance. Its communication distance is lower compared to LoRa, but it is compensated by the possibility to build a meshed network, and then the data can be conveyed from one Sentinel device to another before arriving to the gateway. And in all cases, the data are finally transmitted to the remote server via GSM, the 4G network of mobile phones. The cost of the gateway is quite reasonable, being around 700 euros, including solar power supply. To manage almost in real time the data streams arriving on the remote server, we use a data lake approach based on the one developed by François Thiebaud and other colleagues in the frame of the Neocampus Scientific Interest Group. On a data lake, you have several functional zones. The raw data are arriving on the landing area, where they are recorded without further processing. Then, in the work zone, these data are properly stored in dedicated databases. In this zone, it is also possible to fusion data, to analyze them with machine learning, to generate some alerts if anomalies are detected, and so on. Finally, on the golden area, data are made available to be used by third parties. Practically, how are Econect data processed? Shortly, we first use the Node-RED application to manage data streams. As you can see, this is a low-code environment where we use modular blocks, as in a well-known game for kids. All kinds of data can be managed like this, and no matter they are text or pictures, they are first stored in a MongoDB base as crude data bytes. In a second time, the time-stamped numerical data from sensors are extracted from MongoDB and stored in an InfluDB base that is perfectly adapted to manage time series. The data conversion and transfer from a database to another are done using custom Python scripts. And finally, we developed several graphical tools to display and share data almost in real time by requesting the InfluDB records. So I'm not going to finish my talk with a formal conclusion, as the story is not over. Econect has several other projects in various French regions. I can say is at the origin of another larger nationwide project. Yes, your guess is correct. This is the TERRA FORMA project. TERRA FORMA aims at gathering the expertise of dozens of French research groups to go further than what has been done by each group individually, notably to develop new sensors able to monitor both biotic and abiotic dynamics and to improve their integration in a smart network. Then these new sensors, together with existing sensors, will be co-deployed at 12 sites across France to address socio-ecological issues, as Laurent has presented earlier, related notably to soil and water resources, to biodiversity and landscape integrity, and to the chemical status of the environment. For this purpose, new tools, notably for data analysis, will be developed to monitor integrated trajectories of the territories. I thank you for your attention, and I also thank you all the supports of the Econect project and the different contributors also to this project.